Hello, everyone, and thank you for joining our webinar today. My name is Ellen Chichevsky, and I'll be your moderator. Today's talk is entitled Unpacking Resin Alternatives, Single-Use Purification that Reduces Costs and Accelerates Clinical Journeys. This will be an overview of new research on the promise of Protein A purification devices. Our speakers today are Chase Snyder, Senior Development Associate Scientist at AGC Biologics, and Bill Barrett, Global Product Specialist at Gore. Without further ado, I'll hand it off to Chase to get started.
Thank you, Ellen, for the introduction. Again, my name is Chase Snyder. I'm from AGC Biologics, and today I'll be talking to you about some new interesting research we've done with the Protein A purification devices from Gore. So just as an introduction, who is AGC Biologics? I'll start out with a bit about us. So this slide just gives you a little idea of our different capabilities at AGC Biologics. We're your complete partner and can assist from preclinical through commercial stages of process development and manufacturing. We have a cell line development department. We do cell baking, analytical development, full process development department, as well as helping you through GMP clinical manufacturing from IND through BLA and commercial. We have a wide range of expertise with over 150 products developed, including mAbs, coagulation factors, fusion proteins, and other antibody-like formats. We offer preclinical and commercial manufacturing with variable manufacturing solutions such as fed batch, perfusion, stainless steel, and single-use reactor technology. We have our own in-house Chef One cell line development platform, and we offer speedy DNA to clone selection from 11 months to GMP on specific mAbs that fit our platform. And we work through development into manufacturing and can help you out with regulatory guidance as well. Here's a quick snapshot of our templated platform approach. Again, this is all flexible to meet client needs. The order at which this is done and the timelines are very flexible. AGC is experienced in accelerated development, and we maintain flexibility in meeting your development and manufacturing specifications. Our team collaborates with you throughout process development and scale up to fit your process needs. And we focus on development to optimize manufacturing as well as quality. We have supported scale down and process characterization studies numerous times with success for many different clients. And here you can see our global network of services across 3 different continents. We're able to support mammalian and microbial across these sites, as well as added pDNA support at our Chiba site. Some new key trends that are driving our mAb development work at AGC. Single-use systems are being implemented across a lot of different companies. They offer smaller footprint, less upfront capital. They also allow for you to use less water, chemicals, and energy in terms of turning over manufacturing suites, which allow for more sustainability. Again, they're scalable and flexible, which fit our needs very well. They allow for less downtime and faster turnovers, so we can get more products and more lots through our manufacturing. They also allow for a lower risk of contamination, bioburden, and operational error with a lot of these single-use flow paths and systems. And then moving to supply chain, we understand that it's important to source from vendors with multiple sites of production and work around short lead times. As far as intensified processing, smaller footprint produces higher productivity for us, allows us to maintain our manufacturing capacity in smaller areas. And then our consistent scalability and facility fit allow our speed to tech transfer to maintain processes across different sites. And then working with new complex modalities, a lot of clients are coming to us with new multi-specifics or ADCs or FC fusion proteins, and the molecules are getting more complex as we move along in the future. A common problem still persists, though, and it's downstream specifically, a bottleneck with resin chromatography. And the first step in most of our manufacturing steps is protein A resin for affinity. And these columns are often underutilized, and oversized. They don't really allow us to keep with some of the speed through processing times that keep pace with our upstream productivity. So we need to optimize this chromatography step. We need to optimize the media performance to enable high flow rates and high binding capacities. Along with this step being slower, it is also often a issue for facility fit. So our facilities push through multiple drug products and have different scales with different potencies. A lot of these static columns can be very large and difficult to deal with. They also cause for downtime for packing, qualifying, storing, and validating the resin-based columns, and as well as that the bioburden can often impact bottom line if these storing and validating of these columns for long periods of time doesn't work out as we'd like. With the step being directly after harvest, it is more susceptible to bioburden concerns than other steps in the manufacturing process. So us at AGC thought, what happened if we looked into one of these new protein A membrane technologies and tried to implement that into an entirely single-use process? And so our R&D department did some research to answer this question. We were looking to reduce and eliminate bioburden from storage. We were looking to reduce our manufacturing footprint. We're also looking to reduce potential costs for clinical manufacturing and select scale commercial projects, all while creating a new solution for the next generation antibody-based therapies. So this is where we called Gore to do some research and explore this potential. And this is where I'll hand it over to Bill to speak a bit about Gore devices.
So what we'll do over the next 4 or so slides is just give a little bit about who Gore is, and then just touch on what the technology is, and then what intrigued AGC into testing this out for their single-use application. So next slide, Chase. So Gore's one of the top 200 privately held companies. We have a global footprint across Europe, Asia Pacific, as well as in the United States. And Gore has been in the business for the pharmaceutical industry for over 25 years with various products, having over 50 customer audits performed for the products that we have been supplying to the industry for this time. With the protein A itself, we're seeing an increase in usage, rapidly moving into about 10 preclinical trials with customers since we launched the 1 liter sizes back in March. So as the sizes of the devices grew, we're seeing more interest and a bigger push into the edge of the clinical space. And we have about 8 of the top 10 pharmaceutical companies who have used the technology within their labs. So next slide, Chase. Some of the products supplied by the Farm Bio unit here at Gore include products like our microfiltration media through to flexible freeze containers for bulk drug substance storage, the protein A capture devices, which we'll talk about here, freeze drying trays, silicone-free plungers, as well as some peristaltic pump tubing in our Stay Pure pump tubing line. All of these have been available for various times, as I mentioned, throughout the years. So we'll look now about the protein A product itself. So next slide, Chase. So as Gore looked at the question Chase raised, being oversized protein A columns that are underutilized, we looked at what we could do with our membrane technology and how can we develop one that was scalable and that was easily scalable. And when we thought about scalability, we thought about 3 different aspects, if you will. We thought about the membrane aspect, of course, of speed and capacity. So in the upper left, you'll see a dynamic binding capacity, 10% breakthrough curve, Gore represented in red, and agarose materials represented in gray. And you can see here, again, you wouldn't technically run agarose this fast in any manufacturing type environment, but Gore was getting probably equivalent capacities at 20 or 30 seconds that this agarose material would give you at about 4-minute residence time. So you're looking at 8 to 10 times productivity just on that speed alone when you think about seconds residence time on the x-axis there. It is a combination of a convective diffusive membrane that's partly by design. And so that's why you don't see it as a perfectly flat capacity across all residence times. The second aspect we thought about looking at the base of the triangle was pressure drop. We wanted this to be able to scale and work within the existing capital equipment that's in your labs today. And so we focused on a pressure drop, and we wanted the pressure drop to be well below 4 bar or 0.4 megapascals. So that way it would give you flexibility in that manufacturing environment for how you might want to employ the devices, but be able to utilize existing capital within a good safety margin for pressure. So what you're seeing down there is across 20-second residence time down to about 7.5 seconds residence time, some typical delta column pressures listed in megapascal. The last aspect that we focused on was elution width. We recognize with membranes that some of the technologies out there can give really broad elution width. So we were trying to design ours to minimize the elution width as best we can, recognizing, of course, there'll be differences between a membrane and resin materials themselves. So what you're seeing in the upper right of the slide is the typical elution volume for 100 milliAU to 100 milliAU cutoff for an IgG1. And on average, they would probably be around 2.6, 2.5 CVs. Again, give or take what your elution conditions would be. And that would be something that could be optimized as you did process development further. So in this slide, I think this is where we started discussions both with AGC along the way. So we started off on the left side initially with some pretty low productivity. We were very conservative in the way we were running the devices. And as we worked with Chase and the team at AGC, they were giving different indications around transitions that we could be reducing things. So they started looking at it from more of the operation point of view. And as you go from left to right, you can see each successive change resulted in higher and higher productivities. That included changes of the loading flow rates, all the operational steps, as well as different CIP conditions that we've used. So we've looked across a range of CIPs that you can then tailor to optimize how you'd like to run the device. And these CIPs didn't impact any of the product quality attributes along the way, things like HCP, yield, or the protein A leaching. And then seeing this, I think where AGC got pretty excited was the fact that this would enable them to tailor it to a single-use application that Chase will go into in the next few slides.
Yeah, so I'll pick up off that and continue talking about how we use this Gore device as a single-use solution and how we scaled this up into a 500-liter scale and did some shakedown runs with it. So here's the results from our research. Here's just a basic MAB platform process through protein A, VI, and then your cation anion exchange steps. So again, here's just kind of our limitations we're looking to kind of transcend with this the Gore device. So we will have high volumes in harvest, limited hold times from that harvest, often 24 hours or less. And then when we load these large columns, we often have long residence times, as Bill was saying. 4 to 6 minutes is our average for resin chromatographic media. And again, these columns can be expensive. This upfront resin capital cost is very large. They can be difficult to clean with bioburden issues because, again, this is the step right after harvest. So we're dealing with, I'd say, our dirtiest material in the purification process, and then reuse validation, which comes with using the columns over multiple cycles. So here you can see our Gore protein A membranes. So we used 2 1-liter manufacturing scale devices in this study. We used a 500-liter single-use reactor and processed that through these Gore devices. And we're using a single-use chromatography skid with single-use fill paths for this. So here's our process. It's a basic purification. We perform 19 cycles, switching halfway through to reduce sanitization volume and speed up our cycles a bit. So cycle 1 through 10 was done using the process on the left, and cycles 11 through 19 were done on the right. I would point out to notice how short our residence times are specifically for the loading phase in this case. Again, a standard residence time for protein A resin would be between 4 to 6 minutes, depending on the dynamic binding capacity and the resin that you're using. The rapid mass transfer we get out of this membrane chromatography allows us for a dramatic improvement in the cycle time and boosts operation productivity. And on to some of the output data. So here we have a overlay of absorbance at 280 nanometers for our chromatograms for each cycle. So this is cycle 1 through 19. And you can see that we have really high reproducibility from cycle to cycle, specifically in our elution overlays. These look really good and would not lead us to believe that anything unusual happened. I'd say showing me this, I wouldn't assume that this was a membrane over a column, resin column. And moving on to our pressure trends. Again, over 19 cycles, we didn't see any clogging of the device. There was no increase in pressure during any of this spikes. There's also not any increase in pressure during sanitization when we reduce those sanitization volumes. So as you can see on the left side are overlays of the pressure trends throughout the process. And then on the right side graph, we have a different points in terms of pressure during load, wash, elution, and sanitization. And again, as Bill said, our maximum operating pressure for this device would be 4 bars. And moving on to some of the other data. So here we have lab scale results from our protein A resin we use as a control versus the Gore device at small scale. And then some of the results of that scaled up run in the 500 liter scale that we did. So here, again, that productivity number is a bit shocking, but the increase is quite dramatic. Our average elution volume, as Bill was mentioning, for our scale down column was around 2.7 membrane volumes, and then our scale up was 2.6. In this case, we used the 3.5 mil Gore device, which I don't believe we are looking at as a scale up. I think the 9 mil device would be the smallest device you look at to scale, but this was just our early R&D. So we were using the smaller membranes to allow for a little bit less load material to be used. And then we can see the elution HCP, our log reduction values are very similar between the resin and the Gore device. And this is also kind of standard in what we're looking for in a protein A is about 2 log reduction. Our elution protein A. So as far as the leached protein A ligand, we're looking to keep that value really low throughout the process. And the 2.5 we saw from the 2 liter Gore devices is well within our range. It looks very good. And again, just looking at the average SEC product quality remains pretty constant throughout the scale up of the protein A device from Gore, and it looks a little bit better than our resin column control. So I guess this would be called something of a bridging study between the two products in terms of the resin control and the membrane. So we know when evaluating new solutions at AGC, we have to maintain the same level of quality. We know we're getting this higher productivity number, but we also have to make sure that all of our outputs in the purification process are still similar, if not better. And we can see that the same purification efficacy and product quality output was observed in the lab scale experiments as well as our scale up at the 500 liter. And here's some of extra information and product quality results from our 500 liter run. Again, our residual protein A stays really low. Our step yields are nice and high. The step productivity, again, is nice and high. Our HCP removal off of that first step, again, as 2 LRV brings us down pretty well. And our, as usual, the AEX ends up cleaning that up for us and getting us under the 100 ppm target that we're looking for. The host cell DNA removal is also nice to add in terms of the protein A device. And then again, our SEC output looks good from that first step. So this is all something that we would say, this bridging study is successful. This new device doesn't show any downsides in terms of purification quality, and then gives us that huge upside in terms of step productivity, as well as being a single-use solution for this fully single-use process that we're looking to put forward. So here's just a potential model scaling up from our 500 liter test that we did into a 2000 liter bioreactor. So we're assuming 5 grams per liter. This is a 1 batch examples for some of our, let's say, early stage clients looking to get into the clinic. And you can see on the left side, the cost for 1 batch. In this case, we would use a 32 liter pack bed column. We have, assuming just a 4-minute residence time, this would take us 8 cycles to purify the batch and about 17.5 hours. And then something else that's important to highlight is the preparation time for packing a column like this would take around 32 hours. as well as the storage cost would be around 10% of the resin cost. And then if we look at the Gore protein A membrane column, in this case, to purify the same amount of material, we would use, again, 2 of the 1 liter Gore devices. The cost for the devices themselves is dramatically decreased. Again, our residence time and our set times are much faster. It would take quite a bit more cycles to do this, 157 cycles per batch, but in our case at AGC, we would be using this as a single-use device, so we would not need to store this device or use it again. The preparation time is very low, 2 hours, no storage costs, and our processing time is still similar within that 24-hour period. And so on the right side, you can see the productivity per batch, again, over 10 times more productive on Gore versus our resin solution. And then the column cost of ownership for 1 batch. So if a client were coming in looking to get some material into the clinic to test out a new molecule, they wouldn't need to invest so much in that protein A resin column and could go with this Gore single-use device and look at a 90% cost savings in upfront capital for this. And then moving on to the scalability. And this device is definitely scalable. It's very important when looking into manufacturing and how we can fit things from our lab scale up into our PD scale. I mentioned that we use the 3.5 mil device at our R&D department. If we looked into the 9 mil device at that time and were able to use a little bit more material, I think we would have gotten more similar outputs in terms of that elution CV or membrane volume in this case, and have a little bit better idea scaling up into that. But you can see the portfolio of different sizes that Gore offers here. And then we would mostly be looking at using the 1 liter devices for anything in terms of GMP scale. And they do offer smaller ones for pilot scales as well. So overall, what is the benefit to mAb developers like AGC? So again, higher productivity, 10 times productivity compared to the resin control at both manufacturing and lab scales. This is very useful for us as a CDMO. Faster purification cycles. So again, the entire protein A operation was completed well within 1 workday or 1 shift of 8 hours. So 1 cycle of the purification process was completed in 10 minutes or less, and we did 19 cycles in 3.2 hours. And then the benefit for some of our potential clients could be reduced capital expenses. So we ideally would look at this as a cost-saving measure to allow for more early phase projects looking to get into the clinic as fast as possible. And then in this case, we did use this as a fully disposable upstream and downstream purification. So this allowed us to do something that we had never done before, and is going to hopefully allow us to offer this as a service to clients, as well as eliminating risk and error. So again, column validation testing, chemical consumption in terms of that, as well as cleaning, operator intervention for time to pack and unpack resin-based columns, clean them, and then bioburden associated with storing the columns and the resin as well. That will eliminate all those risks. So separate, we're great, but together we're even better. The promise of AGC's single-use manufacturing technology, this allows us to greatly increase our flexibility and lower clinical and commercial manufacturing costs for customers via single-use technology platform process. And then on Gore's side, their protein A capture device that we're using is single-use in this instance, reduces complexity and risk, and has been demonstrated through multiple different studies to increase productivity and simplify one of the most expensive and critical steps in our protein A process. And then this also allows us, again, to get the best manufacturing yield possible while staying within a well-characterized, validated batch process, and most importantly, keeping our purification product quality high. And with that, I want to ask you to come see us at the upcoming events that we have going on. Both AGC and Gore will be present at these events. And take any questions at this time as well.
Great. Thank you, Chase and Bill. What a great talk. Really interesting approach to purification process technology to address some of these downstream inefficiencies. The first question I have, I'll direct toward Chase. The question is, have you run this process through 2 polishing steps?
Yes, we have. Let's see. Should be able to show. So at lab scale, we did end up pushing this process through a cation exchange step. We did that with a Cytobine S membrane, which is again keeping with our fully disposable process. This step worked well for us. You can see it reduced our HCP ppm down to between 5 and 7. But during scale up, we decided that this step wasn't necessary. It was a bit of a redundancy in terms of purification quality. We didn't really need to reduce our host cell protein any more than it already was, so we decided to leave that out for the 500-liter run.
Interesting. My next question for Bill is related to these membranes. Are these membranes only single-use devices?
Thanks, Ellen. No, it's all dependent upon how a company wants to implement them. So if a company did want to, for example, as Chase showed with a 2x1 liter, if they wanted to go to a 4x1 liter, run it in less time with a slightly oversized, they could sanitize and then store the columns.
Oh, okay, interesting. Next question. Someone's interested in the cost of ownership analysis across multiple batches. Chase, is that something you can speak to?
Yes, I believe we have that. I think this would be the info we have in terms of cost comparison. So in the red, you can see the Gore device as you go per batch. Again, you'll be buying new Protein A devices, but you can see that the break even point at this isn't even reached in 10 batches. And this also doesn't necessarily account for your column lifetime, your resin lifetime. So it depends on what your resin lifetime study would be for the resin based, on how many batches you can actually use that resin for.
Bill, I have a question about the devices here. So Chase was mentioning, GMP scale, lab scale. How do the actual devices scale across different batch sizes?
Yeah, so we tried to keep scaling as simple as possible so that the way we explain it to most users is we scale by residence time. So if you're developing a method, for example, as Chase was saying, if AGC takes a 9 ml as their initial, we're going to use this to set a process and then scale from there. Once you've developed the process on the 9 ml, both residence times, your column volumes that you would use in your protocol, you just port the exact same method and residence times up to the larger scale devices, only changing the flow rates essentially. You do need to account for your system holdup volume, so you would want to make sure you're following that. That's something we learned working with Chase and his team, was to account for some of that as a way to adjust those. But generally, the methods just port up by the residence time that you're going to select, and you're just altering flow rates.
Another question is sort of a more general question, but how might this technology or this process relate to other types of molecules?
Yeah, I can jump in here. So we use this as for a mAb, just a generic IgG1, but we have run a lot of other molecules through the Gore capture devices, and they do work for specific other molecules. And I don't know if you have anything to add on to that, Bill, specifically.
Yeah, we similar to what you said, Chase, we've tested, or not we, but we have customers using it across bispecifics, FC fusions that span low molecular weight to pretty high molecular weight species. IgG, not only IgG1s, IgG4s as well have been tested as well as multi-specifics.
Yeah, we have a multi-specific that binds the variable heavy chain 3 domain specifically, and it also worked with the Gore membrane. So it's not just FC binding. Maybe the one comment I will throw in there, based on the binding capacity curve we showed earlier, that's using polyclonal IgG. So you'll want to check your specific molecule's binding capacity, because as you know, they may have different capacities. So FC fusions might be a little bit lower than the IgG ones, as an example.
Okay. Another question we have is, I wonder, maybe Chase, if you could elaborate a little bit on sort of the significance and the benefit of having a single-use process over continuous processing.
Yeah, so I think it depends on the client's needs. Again, AGC is kind of all about being flexible for different clients and different molecules. A lot of our clients that are looking at single-use solutions have maybe a really high carryover risk, or the molecule is really potent, and we're having difficulty in terms of cleaning. Let's say they were to use a single stainless steel skid or one of the resin columns or something like that. We would have difficulty cleaning the maximum allowable carryover for that. And so a single use would be, in their case, a very good solution to allow their manufacturing to go through our facilities quickly and efficiently.
Sounds reasonable to me. One more question I have for you, Chase, is actually how this process looks across each cycle. So are your HCP clearance or your protein A stable?
Yes, I think we should have some info on that as well. So here, I believe, is our HCP LRV across each cycle. So yeah, the cycles are reproducible. We don't see any, like, let's say HCP leaching or something like that, not binding to the membrane as we have more cycles. This data is specifically from the 500 liter run, but we did do a scale down across, I think it was 60 cycles with the 3.5 mil device. If we were to go back and do that, I think we would use the 9 mil device, as that is the smallest scalable device. So we would redo that to look at the membrane lifetime before we scale up into manufacturing.
Thanks for that explanation. I believe that's all we have time for today. Again, thank you all so much for joining us. I hope you have a good rest of your day, and thanks again to our speakers.